Good morning, students. Hope you will be fine at your respective places. In continuation to our topic, that is the morphology of the flower, today we are going to start the new topic, that is the seed. Now, what actually is the seed? Let us try to discuss that. Seed, it is the fertilizer. It is the matured ovule. Now, what is the seed? Seed is simply the fertilizer. It is the matured ovule. And the study of seeds is known as spermology. Spermology. So what is seed? Seed is the fertilized matured ovule rather, and the study of the seed is called as the spermology. Now this seed has got some parts. Now what are the various parts of the seed? Number first, this seed has got the what we call as this outer covering. That outer covering is called as the coat, seed coat. It is actually the seed coat, and this seed coat, it may be two layered or it may be one layer. This two layered is the mainly found in the dicotyledons. The two layers, what are those? These are outer layer and inner layer. This outer layer is called as testa and this inner layer is called as tegmen. So what we have discussed, this first part, what this seed has got, it has got the outer covering which is called as the seed coat. This seed coat may be two layered or it may be one layered. This two layers is mainly found in the dicotyledons, and we are mainly dealt with this because we are mainly concerned with these flowering plants. And this two layered, it may be outer layer and inner layer. Outer layer is called as the testa and this inner layer is called a tegmen. This is the first. Now second जो हमारे पास है, that is the endospermal axis. What is the endospermal axis? If we happen to what we saw, we remove the covering of the seed, what we will find? We will find this slide structure. This is called as the embryonic axis, this endospermal axis. This is called an endospermal axis, but this is not too much thick. It is very much minute, and it has got these two. In case of the dicotyledons, these two semicircular structures. These circular structure, not semicircular, but these structures which are called as cotyledons. These are called as cotyledons. The point at which these cotyledons and this cotyledons at the point at which these cotyledons they meet with this endospermal axis, that is called as this cotyledonal node. Now this part, that is towards the chalazal and this is the micropylar end. Towards the chalazial end, the tip of that, this part, this tip, that is called as plumule. This plumule, it gives rise to this stem, and the tip towards this micropylar end, that is called as a radicle. This radicle gives rise to the root. What are these micropylar and what are the chalazial end? This will be studied when we are going to discuss the structure of the typical ovule. Now there is one more part in this endospermal axis. What is that? This plumule and this cotyledonal node, we have got this structure, this greenish structure, that is called as epicotyle. And similarly, between this cotyledonal node and this notch and this what we call as the radicle, that is called as hypocotyle. So this is the second part. That's the endospermal axis. It has got these two parts, and these different parts, they also give rise to the seed. Now there is the third part. What is that called as? That is called as the reserve food. This reserve food, it may be cotyledons, or it may be endosperm. Cotyledons, अगर हम बात कर लेंगे, either the number of cotyledons, it may be one, or it may, when there are the one, that is called as monocot. When there are the two, then it is called as the dicot. Or what we'll see, it is the endosperm. What is this endosperm? Endosperm structure which is formed by the triple fusion during the process of double fertilization. That you will study in what is the double fertilization, what is this in the embryology portion, where it will get altogether clear. So there are the three parts of that is the covering, and this covering as seed coat, what we will call as, it is two-layered. There are two layers, outer layer and inner layer. In two layers, it may be the two-layered, or it may one layer. What we call as this two layer, it may be either outer layer and inner layer. The outer layer is called as testa, and the inner layer is called as the tegmen. Then we have got the endospermal axis, and this endospermal axis has got these parts, and then we have got the reserve food, and the reserve food may be either in the form of cotyledon, or in the form of endosperm, or in the form of both cotyledons as well as in the endosperm. Now in order to understand the structure of the seed, what we have studied here, the chalazial and the radicle, and let us try to discuss, let us try, in order to make the things more clear, let us draw the diagram of a typical ovule. Diagram of a typical ovule. This is the stalk. This is the stalk of this. This is the stalk of the ovule, which is called as funicle. This is called as funicle. And this is the body of this, what we call as the ovule. Now what we will see here, what are these? These are the integuments. Now this stalk is attached, this ovary, it is attached with this ovary, and here what we will see is the placenta, and this placenta has given rise to this ovule. This is the typical dicotyledon ovule. From the diagram, this is the integuments. This is the outer integument which gives rise to this testa, and this is the inner integument which gives rise to this tegmen. Now, what we will see here again, now from the diagram, it seems as if these integument, they arise from this end. Where from they arises, it is called as the chalazal end, which we have seen there. And this pore-like structure at the below, it is called as micropylar end, which we have shown here. Now, this stalk अगर हम बात कर लेंगे, it is attached, जो हम पर एक है, there is this stalk with this ovule bar, this I will draw with this another this color, this the reddish color मैंने बनाई यहाँ पर, and this is called as this, what we call as, this is called as hilum. This stalk, it separates it from the ovule. Now, in sometimes we will see that there is the extension of this funicle, but we will see extension यहाँ पर हो जाते, and it arises over this outgrowths हम यहाँ पर देखते हैं on the body of this ovule, and that is called as raphe. This is an extension, and this extension outgrowth which happens to be on this man ovule body, this is called as the raphe. Now, now this hilum, it separates this funicle from this ovule, from this main body, and when we draw the diagram of a typical dicot seed, for example, I will give you the bean. Bean के अगर हम diagram देखेंगे, it has got this, what we call the kidney shaped diagram. Kidney shaped you will find there, and this, what we will see, you will find some scar shaped structure on this concave side, and if we go through this concave shaped structure, we will find here this pore, then this scar, then this one more scar. This one, this pore, this is called as micropylar pore. This is this micropylar pore. Now this scar जो यहाँ पर है, it is the hilum scar, and this is the raphe scar. So all of them, they are found in a what we call as in sequence: micropylar, then hilum, then this raphe. Micropylar pore, then there's a hilum scar, then these are those structures which are somewhat naked, which we can see with our eye, and these are found at this point on this concave side of this bean plant. So these different structures जो मैंने ovule के बताए, they give rise to the seed, and they form the different parts of the seed. This was a little bit introduction about the seed. In the next topic we will proceed further. Until then, goodbye. God bless you.